Friday, we went through the mechanism of transcription, and today we're going to start to look at the three major classes of RNA that are found in the cell. These are the major classes. There are probably several dozen minor classes that will only be mentioned very, very briefly. But before we can look at the RNAs that are being made, we have to understand a little bit about how we designate and talk about the RNAs. So most of the RNAs in the cell are described in terms of their size. And there are two very different ways to talk about the size of a large molecule. One is like P53 that we talked about earlier, which stands for a protein of 53,000 molecular weight units. And so putting that unit designation on is a very nice way to talk about small molecules. However, once we get up into large molecules, we're starting to talk about millions of molecular weight units. It doesn't work very well. So instead, S units are commonly used. So most people think that S stands for sedimentation. How fast a molecule can sediment through a sugar gradient, a sucrose gradient. A little bit different than the cesium chloride gradients we talked about. And we are not going to go through the method of how this is determined, but just I want to describe to you in very brief details what this is all about. S does not stand for sedimentation, even though that's the method used. So a large molecule will sediment through a sucrose gradient faster than a small molecule. S actually stands for Stedberg, who is the guy who originally described this system. S units are logarithmic units. So once we start looking at details, those details are going to be increasing in a logarithmic fashion, not in a linear fashion. So we're going to use this designation to talk about the major classes of RNAs, especially the larger classes of RNAs. Any questions on that? So what we are going to look at are, first of all, the ribosomal RNAs, where little r stands for ribosomal. tRNA, where T stands for transfer, and mRNA, where M stands for message. We've already talked very briefly about this when we talked about how the code was broken. We went through very, very briefly the function of these different molecules. We're going to look at that in more detail in this chapter. If we look in a cell and the amount of RNA, what we find is about 75% of the RNA that's in any cell, just cellular RNA. Doesn't matter whether it's a human cell or a bacterial cell. This is pretty much the universal amount of ribosomal RNA. Transfer RNA is about 20%, and messenger RNA is about 5% or less. Actually, 5% is pretty much closer to the top limit, but just rounding off numbers. And so what we see is the vast majority of RNA is ribosomal. But we get a different picture if we look at the number of genes that code for these RNAs. So if we're talking about bacteria, what we see is about 7 ribosomal genes. The tRNA is about 50, and the messenger RNA is about 4,000. And so those are, again, rounded off numbers, but the numbers come from E. coli. And so the important thing here is that these percentages are virtually invariant. There is virtually no significant difference between the proportion of these RNAs in any bacteria or any eukaryote cell, plant or animal. But what differs is the number of genes. The number of genes scales with the size of the genome and the complexity of the organism. So these are bacterial numbers. If you go and look at a small eukaryote like yeast, just multiply by 5 or 10, and you'll come up with lower eukaryotes. Plants are outliers. Plants have a lot of genes because so many of them are polyploid. But then this number will scale with the complexity of the organism. So what we're going to look at in this chapter are the genes that code for these 3 RNAs and what these 3 RNAs are doing in the cytoplasm in terms of gene expression. So any questions on that before we start to look at the ribosomal RNA genes? We're going to do this back and forth between bacteria and eukaryotes. So we're going to start off bacteria, ribosomal RNA. The function of ribosomal RNA is to make up about half of the mass of a ribosome. Remember from the code, breaking the code, the ribosome is the factory that makes proteins. Transfer RNA is the molecule that transfers the subunits, the amino acids, to the factory. Message is the blueprint. So ribosomal RNA, we're going to start with the RNA, and then we're going to go on to the genes. Ribosomal RNA makes up a ribosome. A ribosome, a generic ribosome, that's a very bad cartoon. It's not bad art. It's actually really nice art. But in terms of the reality, it's a bad picture. This is the classic picture that's been used since the 1960s. But we now have exquisite detail on the structure. And so the old view of a ribosome, if you get any older books, is like 2 basketballs squished together. A small basketball on top and a larger ball on the bottom. The reality is the ribosome is a donut. And there's no way that I'm going to draw a 3-dimensional donut. And so we're just going to have to be satisfied with this very bad representation of what a ribosome is. And so we talk about a small subunit and a large subunit. We're doing this in bacteria first. So in terms of the subunit, the small subunit is 30S, the large subunit is 50S. So now I'm using these S units because these are really large particles. I'll break down what's inside a small subunit and what's inside a large subunit in very general features. The ribosome, so the 30S and 50S, gives a ribosome that is 70S. So 30 plus 50 add up to the entire ribosomal unit as a 70S ribosome. These numbers are going to be slightly larger in eukaryotes. The sort of rule of thumb is that for no apparent reason, pretty much all the molecular machinery that we look at in bacteria will be larger in eukaryotes, even though your ribosomes are doing virtually exactly identical reactions to what's described for the bacterial system. So even though they're doing the same, your ribosomes, your ribosomal RNA is going to be larger. Any questions so far? Yes?
Why isn't it 80?
Ah, thank you. I was hoping somebody would ask that question. See that word log up there?
Yeah.
Yes, it's adding logarithmically. I knew somebody was going to save me and ask that question. Does everybody understand? We're adding in a logarithmic scale. And so every time we do that, things are not going to be proportionally different, but they're going to be logarithmically different. So now we can start to break this down and see what is in a small subunit. And there's going to be a vocabulary problem here that I'll tell you about. And so in this small subunit, we have a 16S ribosomal RNA and a 23S ribosomal RNA in the large subunit, plus a 5S ribosomal RNAs. Plus about 50 proteins. So our focus is going to be on the ribosomal RNAs. 3 different ribosomal RNAs make up the ribosome. That's not going to change. The overall picture of the eukaryote ribosome will be nearly identical. Just the numbers are going to be larger when we move over and do the ribosome in eukaryotes. And so the terminology problem is that this is the small subunit and this is the large subunit. So the 16S RNA is very often referred to as the small ribosomal RNA. And you immediately see the problem with that because very definitely 5 is smaller than 16. The problem came from the fact that the 2 large, the 1623, were named before anybody found the 5S. And so this is called the small ribosomal RNA. This is called the large ribosomal RNA. And then we have a 5S, really small RNA in the large subunit, plus a bunch of proteins that we're not going to go into the details of. So that is a ribosome in bacteria. Its function is to decode the message and to make polypeptides, to put together amino acids. We'll go through that mechanism in due course. Any questions about a bacterial ribosome? The major components, we are focusing on the RNAs, not the proteins.
What is the red arrow or whatever it is through the 23S?
No, that's just, so there's a 23S ribosomal RNA and a 5S ribosomal RNA. That's all I meant. And a 16S ribosomal RNA. So 3 ribosomal RNAs, but the small ribosomal RNA is not the smallest ribosomal RNA.
So the 23 and the 5, do they make up the large RNA?
The 23 and the 5 plus most of the proteins make up the large ribosomal subunit. There's another hand here.
What's the first thing, the left numbers and the right numbers? 30S and 16.
Oh, so 30S is the size of the whole subunit, which includes the 16S RNA plus a bunch of proteins. Well, I don't want to break down the proteins and the subunits. We're focused on the RNA. Just understand that the RNAs aren't by themselves. They're complex with proteins. Anybody else before we move over and look at your comparable ribosomes? Okay, so that's bacteria. In eukaryotes, it looks the same. For all intents and purposes, the ribosome looks the same, it's just slightly larger. No, they're going to be larger, and so.
Oh, no, like for all eukaryotes in general, like are yeast and us have the same size?
Yeast and us pretty much have the same size. Us and plants. This is pretty universal. There are obvious small differences between species, but nothing significant. This is the universal pattern, same as true in bacteria. Universal pattern. And so now we have a 40S and a 60S, same thing, adding to an 80S ribosome. So each subunit is larger, and that means that the ribosome itself, 70S and 80S, your ribosome is larger than an E. coli ribosome. The 40S has one small RNA. It's now 18S. The 60S has a 28S. The 5S is the only thing that's the same size. Plus 50 proteins here, plus about 75 proteins. So larger ribosomal RNAs, larger subunits, more proteins. But the function is exactly the same. Any questions on the introduction to what is a ribosome before we look at the genes that give rise to these? There are only 3 ribosomal RNAs in each species. Yes?
I wanted to ask, like, what was the definition of a ribosome? Like, the function, like, it decodes the message of DNA?
Exactly. So, I would say more generally, it decodes the message and it makes the peptide bond. So it's the factory that makes the protein. That's what a ribosome is. It's the factory that makes the protein. 2 components. You have to decode the blueprint, the message, and you have to synthesize and put together the amino acids to make the protein. Anybody else before we start to look at the genes? So again, we're going to start off with the bacterial system. And so the way we are going to do this is to look at 3 different aspects to the genes. And this is going to take weeks because we're going to go through all 3 genes, all 3 classes. So we're going to look at gene organization. We're going to look at gene structure. And we are going to look at gene regulation. And so this is the system that we are going to use for a couple of weeks to go through the ribosomal RNAs, then we're going to do the transfer RNAs, then we're going to do messenger RNAs. Gene organization and gene structure, you already have a handout weeks ago that focused on the messenger RNAs. And so the definitions are exactly the same. Gene organization means how multiple genes arranged in the chromosome. We only have gene organization if we have multiple copies. A single gene can't have an organization. It is. But only for multiple copies. And the ribosomal RNAs are going to be multiple copies. Gene structure is what a gene looks like. Gene regulation is how do we turn the gene on and off? How do we regulate the synthesis of the RNA from the gene itself? As I said, we're going to spend several weeks going through structure, organization, and regulation of a bunch of different genes. So it's going to be a major question on the third exam right before Thanksgiving. So you want to make sure you understand the difference between gene organization, gene structure, and gene regulation. If you give me a perfect answer for gene organization, but give me the gene structure, you won't get any points. So this goes back to the handout. You have a general handout on gene structure and gene organization, what mRNA genes look like. Any questions on this? I would recommend making a spreadsheet. Set it up just like this for all the genes that we're going to go through. We're going to go through the major classes of genes. So let's start off. We're talking about ribosomal RNA. We have 3 different ribosomal RNAs. We're talking about this in bacteria now. So gene organization for a multiple copy gene, we're talking about bacteria. We could either have multiple copies scattered, or we could take the same number of genes and do tandem. Those are the only two systems of gene organization that we're going to look at. I don't think there's any others. Does everybody see the difference between an organization that's scattered? We have multiple copies. We can put them in the chromosome, either spread them out, or we can put them all together. Any questions on what I mean by scattered or tandem? Yes?
With scattered, do they have to be equidistant, or does it matter?
No, if they're not all together, you can have, no. So anything that you can draw on paper probably exists. And so you want to scatter them irregularly or regularly if they're still scattered, if they're not. And so tandem is a really tight definition. That means side by side by side by side by side. And we're going to see both. Does anybody, so this is critically important. I mean, we're going to use this over and over and over again. Any other questions on what I mean by these 2 different modes of organization? And so we're talking about bacteria now. I should have counted more carefully. So gene organization, this is specifically E. coli. That's the lab rat of microbiology. We've done a lot. We're going to do more in E. coli. And so gene organization, first of all, multiple copy. The common laboratory strain, and there are different strains of E. coli that can differ considerably, the common laboratory strain has 7 copies. So this is multiple copy and scattered. Remember now, we have 3 different RNAs. And so the organization all three in one transcription unit. So all three RNAs, 18S, 23S, and 5S, are all in one gigantic, for bacteria, transcription unit. We went through transcription unit on Friday. Well, that's the organization. It actually looks just the way I have it in that model. They're almost perfectly spread out, but that doesn't matter. They're just not tandem. Any questions on gene organization, bacterial gene organization? This is the typical pattern. So now let's look at gene structure. Gene structure means what does one of these genes look like? What's the structure of a single gene? And by the way, all 7 are absolutely identical. Absolutely. So the structure now promoter to terminator will let a box represent the transcription unit. I'm not going to give you the details of the promoter because we did that on Friday, but this is a standard E. coli promoter with a -35 box and a Tata box spaced out exactly the way we did it on Friday. And so what we have inside is coding sequence for 18S. If I do this right, 23, not 18, 16S, 23S. If I set that up right, 23S is almost exactly twice the size of 16S. And then we have, and so this is drawn way out of scale, because 5S is tiny compared to 16S, but I want to make sure you see it. So this is not drawn to scale. And so I have these hatched boxes in this giant transcription unit, and then we have white space. So the white space is transcribed spacer. So separating everything out, we have transcribed spacer. Transcribed spacer just means a sequence of DNA that is copied into RNA and then cut out and literally thrown away. There's one kicker here, and that is inside some of the transcribed spacers, it differs. We have tRNA genes. So this is a mixed-use gene. Every single one of E. coli's ribosomal RNA, it's called ribosomal RNA, even though there could be several different transfer RNA genes, it's still called a ribosomal RNA. It just means you only have to learn one structure and you cover 2 different RNAs, 2 of the major RNAs. Any questions?
So not all of the transcription spacing is identical?
That's correct. So some of the transcribed spacers will have an extra, it's actually a really tiny gene compared to the ribosomal, an extra tiny gene in them, but not all do. That's correct. Just the way I've drawn it. Anybody else?
What does P and T stand for?
Promoter and terminator. So we covered that on Friday. You have the structure, the sequence of most of the promoter, but not the terminator. Anybody else? Any questions? Yeah.
So does the transcript space always have tRNA, or is that just...
Nope, just like I've drawn it. Some do and some don't. Anybody else? Any questions on what is a bacterial ribosomal RNA gene? What you're looking at is the answer to what is a bacterial ribosomal gene structure. So this RNA, this gene rather, is transcribed into an RNA. That came out pretty good. So what you see is a transcript, 5' to 3'. Here is what will be 16S RNA. Here is a tRNA. Here is 23S RNA. I'm going to raise the board in just a second. Here is 5S RNA and another tRNA. You don't need the top anymore. So the way that we get a ribosome, we transcribe this gene, here's the promoter and terminator, into a very large precursor RNA that has all these goodies inside, and then we process, that is, we cut out 16S plus 23S plus 5S plus tRNAs. So processing, and we throw away, we let the transcribed spacers just degrade. We throw away the transcribed spacers. They just help in folding to get the RNAs folded right to make that big ribosome. So processing is all about cutting out the RNAs and folding them up right. The tRNAs fold up automatically. The ribosomal RNAs fold up while they're being complexed with those ribosomal proteins. Any questions on bacterial ribosomal genes?
It folds into the ribosomes. So yes.
I'm sorry?
It folds the RNAs into ribosomes.
It folds, well, so it folds the RNAs so that when they bind to the ribosomal proteins, you end up with a ribosomal subunit. But yes, now the folding plus the proteins binding is what gives you the ribosomal subunit, each one. And they fold up, in general, about the same way. Any questions on ribosomal RNAs?
Spacers are what makes them fold.
No, well, the spacers help the folding process. It doesn't make it. So it's the internal structure of the ribosomal RNA, the 23S, the 16S, that really dictate the folding, plus the proteins that associate with those regions. So the ribosomal proteins are reading the RNA to bind properly.
So if we had that unit, the 16S would sort of collapse down to 23, 5.
No, I don't want. It's complicated. It's being processed actually while it's being transcribed.
Do the tRNAs follow?
don't even want to go there. It is being cut up and folded during transcription. So it gets a little complicated, but it's not necessary to go there here. Yes?
Will you repeat what processing is?
So processing is cutting and folding. Cutting and folding, we got to cut out those borders of all the transcribed spacers, and they get thrown away. Anybody else? Any questions? We have one more major topic that's relatively simple.
So then the 16S, 23S, and 5S are going to go bind to the proteins to make the ribosome, and then the tRNA is just
They're
off
off,
the
they're
cytoplasm.
off on their own. They do their own thing. So remember, there's no nucleus in cytoplasm. We're in a bacteria.
Okay.
Anybody else? Okay. Gene regulation. Simple and straightforward. This is all about growth control. We've talked about growth control in eukaryotes. It's pretty much the same thing. Bacteria especially, if they have enough energy, they're going to divide. It's just that simple. So growth control in bacteria is pretty much what's the concentration of ATP. So if there's a minimum threshold for ATP, and you cross that threshold, go higher, that will trigger the cell to get ready to divide, which means it has to double everything. You've got to double the number of ribosomes. You've got two daughter cells, double the number of tRNAs, double everything. And so this is all about growth control. Yours is going to be the same way. This is going to be handled somewhat differently. If you think back to some stuff that we've done, you can get some notion of where we're going to go with that. So growth control in bacteria is relatively simple and straightforward. Growth control is all about having the right concentration of ATP. Any questions? Yeah.
So the growth control, is that an all or none then?
Yes, it is. It's all or none. You either have enough ATP and the cell starts to get ready to divide, which means replicate, double everything, or else you're just going to sit there and wait until times gets better. Anybody else? So that's E. coli. Now we can look at eukaryotes. So if you think about this gene organization and gene structure in E. coli, it really makes a lot of sense. If you want to make a ribosome, you just transcribe a gene, and you've got your 3 ribosomal RNAs just sitting there. All you got to do is make the proteins. Unfortunately, you decided that that wasn't very good. You wanted to do things differently. So we're going to have to divide your ribosomal up into 18, 28S, and 5S genes. For some reason, and this is universal eukaryotes, as eukaryotes split off from bacteria, there was a major alteration in the ribosomal RNA genes that really makes it difficult. You have to go through some really very interesting regulation because you want to balance off these giant 1828S genes with these little teeny tiny, this thing is drawn way out of proportion, little teeny tiny 5S genes. You really need to balance it so you don't waste energy and make a lot of 5S RNA for which there's no 1828S. But we're not going to look at that detail. So again, we're going to look at gene organization, gene structure, and gene regulation. So again, let's just number these. Yeah, they are 1, 2, and 3. So number 1, 1828S, we have multiple copy, just as in bacteria. Remember what I said is that the gene number scales with complexity. So yeast, so E. coli has about 7 genes. Yeast has, I don't know, about 100. Drosophila has a couple of hundred. You have about 400 or 500. So it's multiple copy, just generic for eukaryote. But now the huge difference is your 1828S genes are tandem. We're going to go through and do 1828S, then we'll do 5S. So your genes are arranged tandemly in a very special part of the nucleus called the nucleolus. It was named this way back before anybody knew anything about ribosomes and ribosomal RNAs. If you look in any eukaryote nucleus in interphase, what you see is this grainy material that represents the chromatin that's opened up, and you see typically one large region, maybe accounting for 15–20%, pretty obvious, that's a very dark, grainy material. And that's the nucleolus. It's dark and grainy because, oh, I erased it already, because 75% of the RNA is coming from just a few hundred genes. And so transcription is typically very, very heavy, especially in a dividing cell in the nucleolus. Not only is the nucleolus the site where the ribosomal RNA genes are, it is also the site of assembly. So I said that this is happening in bacteria while it's being transcribed. It's not the case with yours. You're going to make the complete RNA and then go ahead and process it and make a ribosome. So the organization is multiple copy, tandem in the nucleolus. 18 and 28 have split off from 5S. We'll do 5S soon. Any questions on what I mean by that? Then I'm going to draw out what tandem copies of these genes look like. So number 2 is gene structure. So a tandem set of ribosomal genes not drawn to the same scale as over there. A similar picture, we have now 1828S as opposed to 1623S. We have transcribed spacers proportionately about the same size. Now, though, only because we have tandem copies, we can now talk about non-transcribed spacers. The DNA space in between these hundreds of copies of ribosomal RNA genes. And so that's what this represents. This just goes on and on and on and on. But other than that, very similar. Just take out that one last section of 5S and never any tRNA genes. These are just pure 1828S transcripts. So a very similar but not identical story. Any questions on that? So this, hang on, this is This is transcribed. And then processed. The big picture is the same, but the details differ because we wait until the whole thing is made before we start processing. Someone had a question over here?
What does the ETS stand for?
What is the non-transcribed spacer? So now, because of tandem genes, we have transcribed spacer inside the TU, and we have non-transcribed spacer in between the transcription units. The processing is very similar but not identical. So the ribosomes are made here, which means we're in a eukaryote. We have to make the ribosomal proteins out in the cytoplasm, bring them into the nucleus, assemble them into a ribosome, and send the ribosome out into the cytoplasm. We're going to make the 5S RNA way out somewhere else in the nucleus, and that has to be transported into the nucleolus. So because you separated out 5S genes from 1828S, and because you have a nuclear membrane, assembling a ribosome is quite a different feat in eukaryotes, much easier in prokaryotes. Any questions on the 1828S genes? Three is still gene regulation. It is still growth, but now we have growth factors, cell cycle, the whole story that we did on the regulation of the cell cycle, the signal transduction pathway kicked off by growth factors, that now controls transcription of these genes. But the bottom line, the rationale is still the same. If your skin cells or muscle cells or nerve cells are not replicating, these organelles are pretty much immortal. Your ribosomes don't die. Only time you need to make more ribosomes is in stem cells now that are undergoing cell division. But these are very, very stable, nearly immortal structures. Any questions? Yes?
Is there also tRNA in between?
No tRNAs anywhere near these genes. No tRNA anywhere near these genes. That's another major difference. So we have tRNAs and, this is 5S, neither of these are present in your 1820. So your 1828S are only 18 and 28S RNAs.
What did you say the blue near the underneath tandem?
Oh, so the nucleolus, the ribosomes are assembled in the nucleolus. So the nucleolus is not only the site of synthesis of 1828S RNA, it's also the site where ribosomes are put together from all the components. Yes?
What is the thing in red say
Transcription.
transcription?
Yes. So we do transcription and then processing. Make a gigantic RNA and then cut it up. It basically is the same as in bacteria. We make a large transcript and then cut it up. Cut it down to the pieces, the parts that we want. Any questions? Okay, so what's important here, remember that you have 3 different RNA polymerases. Bacteria has one RNA polymerase that does everything. So this transcription is done by RNA pol 1. You have specialized polymerases that handle different promoters. Bacteria has one RNA polymerase. Any last questions on 1828S before we do 5S? All right. So again, 5S is multiple copy and tandem, but as opposed to the nucleolus, this is now just out in the nucleus. Five S are really small genes. But everything here has transcribed spacers. So we saw transcribed spacers here in the bacteria. We saw transcribed spacers in the 1828S gene. Even these little 5S genes, and I mean they're little. The 5S RNA is 120 nucleotides. The 18S RNA is about 2,000 nucleotides. 28S is about twice the size, 4,000 nucleotides, compared to 2,000. And so that's a tiny little RNA. These things are not being drawn to scale. So we have transcribed spacers, and we have RNA pol 3, not 2, but 3. So multiple copy, tandem, and that's what the gene structure looks like. Pol 3 is a very odd polymerase. All the polymerases that we've seen so far, RNA pol 1 and the only bacterial RNA polymerase, have a standard promoter. RNA pol 3, the promoter is inside in the middle of the gene. RNA pol 3 promoters, the vast majority, are internal promoters. We talked on Friday about how your RNA polymerases, all of them, all 3, are illiterate. All 3 polymerases require transcription factors to pre-bind. And so the same thing happens here to pol 1. Transcription factors bind here, and then the polymerase come in. Here, transcription factors bind in this whole region. It's a small gene. It's not drawn to scale. And the RNA polymerase still gets it right in terms of where it sits down, so it's at the start site of transcription. So this is gene organization, multiple copy tandem, and the picture is the structure of a 5S gene. Remember, to make a ribosome, have to transcribe this. We have to process it. And then we have to take this and transport it into the nucleolus to be assembled into the large ribosomal subunit. We still have to cut off the transcribed spacers. It folds up automatically and is escorted from its site of synthesis into the nucleolus by a protein. Any questions on 1 and 2? Yes, and that's really important. That's a good point. Thank you. So each one of these is an individual transcription unit. Each one of these boxes has its own promoter. There's no read-through here. That's really important. I'm sorry? Oh, absolutely. Yeah, T, I didn't put it here. Each one has its own terminator. Absolutely. So there's no read-through. Each little box is transcribed individually.
So is the 5S processed in the nucleolus?
No, it's processed in the nucleus, which just means, just means trimming off those end tabs, those transcribed spacers.
All right, so then with that, it gets transported into the nucleolus, being transcribed into the large?
No, inserted, not transcribed. Transcribed is RNA synthesis. It's going to be inserted into the large ribosomal subunit as the large ribosomal subunit is being assembled. Remember, the proteins have to come in from the cytoplasm.
So do each of those units like code for individual 5S's?
Yes? I cut you off, but was there anything more important?
Not really.
Okay.
So these ones are tandem.
Yes.
How are they different in gene structure than the scattered?
Oh, so structure doesn't matter whether they're scattered or tandem.
Oh,
It's just how they're organized. That's the only difference, yes.
so those ones tandem meaning they're just
But,
surrounding
so both of those sets are tandem.
Yeah.
So the 1828S are tandem, the 5S are tandem. We haven't, the only scattered ones are these here in the bacteria.
And that's just scattered along the chromosome.
Well, certainly, yes, around the chromosome.
But so close, there's still going to be the space in between.
Yeah, but so now they're huge spaces
Yeah.
because it's a big chromosome.
That says 120 nucleotides for the 5S.
Yep. Yes.
And this spacing between the 5S one, or is that the non-transcribed space?
Spacing is probably drawn to scale. The thing is that this is not drawn to the same scale as this. So those are tiny compared to this, which is about 8,000 or 9,000 nucleotides, 10,000 nucleotides. And this is maybe 150 nucleotides. So way out of scale. No, the spacers do not code for anything. That's correct. But everything has it. I mean, all the way from E. coli with transcribed spacers, 1828S, the 5S, and we will see tRNAs all have spacers, transcribed spacers. Anybody else? Yes?
So the non-transcribed spaces and the transcribed spaces.
Right. Because they're tandem, there's also the same non-transcribed spacers. I saw a hand up over here.
Yes. About the, like you have the different units, the arrow coming down, what does it say there? So it looks like a T something
Here?
up higher. Maybe. So right. The one with a bunch of lines.
Here?
Yeah,
Transcription.
okay.
So coming off the gene, that's the only way to get the RNA is transcription. I use a lot of abbreviations, I'm sorry.
So the prokaryotes have tRNA sometimes scattered?
Listen, we're gonna do tRNAs in just 2 minutes, yes. Anybody else? Any last questions? What we have left now is 3, which is the same here. It is growth and growth factor signal transduction. So among the things that happen in signal transduction is lots of genes are turned on to get ready to duplicate and make a second copy of a cell. Any last questions on ribosomal RNA genes?
For the 18 and 28S, number 3, it's growth factor. Does that include, that's the signal transduction?
That is the signal transduction pathway. Yep, absolutely. Anybody else? Okay, so now we can look at the genes that code for the second largest group of RNAs, tRNAs. So as I had in one of these cartoons, the structure of all tRNAs is the 3-leaf clover. tRNAs universal. 4S, not 8. It's a 4S, so slightly smaller than the ribosomal. It's around 75 nucleotides in size. So this 3-leaf clover, these arms are all hydrogen bonded together. The amino acid gets covalently attached to the 3' end. So 5' there, 3', 5'. The function of the tRNA is to decode a codon. The function of a tRNA is to decode base pair with the codon on the message. So here I'm starting off. We have an AUG start codon. This is 5' to 3'. What we have here, we have anti-parallel base pairing, just like we do for replication, just like we do for structure of the double helix, just like transcription. So the codon is 5' to 3'. The anticodon is 3' to 5'. Close to, but not exactly standard base pairing. We're not going to go into that. This is the function of the tRNA, is to bring an amino acid into the ribosome that's being made from the components on the top board. This is how it functions. We'll look at this in more detail at the end of this week, beginning of next week. Decodes the codon. So something has to read the codon and bring in the amino acid. That something is the transfer RNA, which transfers the amino acid to the ribosome. Anybody else? So we have the exact same thing. We're just going to do this over and over again. We have gene organization. structure and gene regulation, made much easier because we have the template on top for ribosomal RNA. We are now on this side in bacteria gene organization. Again, multiple copy. tRNAs are absolutely essential. You can't have a single copy. If you lose one and the whole cell dies. So these are multiple copy, but they're low multiple copy. So bacteria have somewhere around 75 to 100 genes. So multiplicity is somewhere around 1 to 4 copies of each different tRNA. Remember, you've got 61 coding codons, but you don't need that many copies. You don't need that many tRNAs. Gene structure. So in the ribosomal RNA genes, I'm not going to draw anything out again. I've already drawn one of the gene structures for bacterial transfer RNA. They are also in some mRNA genes. So these little guys get around. They are co-transcribed with ribosomal RNA, all the ribosomal RNA genes. They are co-transcribed with some messenger RNA genes, plus singles and doubles. So a single means, looks just like the 5S gene in eukaryotes. It's just a small little transcription unit with one tRNA. A double means a transcription unit twice the size with 2 tRNA genes in one transcription unit. Not going to bother to draw them out. You just have to know that these are multiple copy and scattered all over the place. Growth is very simple. The same rationale we had above. We only need tRNAs, just like ribosomal RNAs, when we are dividing the cell and we have to double all of the contents. This is the only one of the major RNAs where the tRNAs are identical. Notice the difference in size, difference in almost everything for ribosomal. You can take your tRNA set out of one of your cells, drop it in E. coli, and they'll work just fine. You can replace your tRNAs with a set of tRNAs from E. coli, and they will work just fine. This is the only major RNA that you can say that of. So the tRNAs are not different like the ribosomal RNAs. They are virtually all identical. Give or take a few bases, around 75S, they are all 4S RNAs. Oh, let me just give you one other number. That corresponds to about 25,000 molecular weight units. So that's the conversion. Just remember the S units scale logarithmically, so it's not a linear relationship. So this is a universal picture of the function of tRNAs. This is how tRNAs function. Simple and straightforward once you learn the ribosomal side. Any questions on that? Yeah.
I can't remember that says there on the bottom of the mRNA, in RNA genes.
Oh, plus, so the genes for tRNAs are also found as a transcription unit with one single copy, and transcription units with 2 copies of tRNA genes.
Single what?
Transcription units. Yes?
What's written in
amino acid, AA, my abbreviation. Anybody else? Louder. Yes. Yeah, right. So they're just all over the place. Some of them are co-transcribed with other things. Some of them are transcribed just by themselves. Anybody else? Good, we have a few more minutes. So gene organization. Multiple copy. Again, they scale with complexity. So bacteria typically have less than 100. Eukaryotes have hundreds. Yeast have fewer than Drosophila. Humans have more than Drosophila. So hundreds, not quite up to 1,000. They are, now this is tricky, scattered in mixed clusters. So what do I mean by that? Let me put this out here. So there's a chromosome. We have a cluster of genes there. We have a cluster of genes there. A cluster of genes there. So we can take this cluster and look at the cluster. So that your tRNA genes are in scattered clusters. The cluster here is mixed. So we have gene, we have gene, we have gene, gene. This is RNA pole 3 again, just like the 5S, which means promoter in the middle. Each box is a separate transcription unit. Notice that you have two different kinds of tRNA genes. We have the standard gene, and then we have a gene split by an intron. So, gene organization, multiple copies scattered in mixed clusters. Here's the picture. Like all the others, we have transcribed spacers. Just like all the others we have considered so far. Any questions on what is a tRNA gene? When we come back on Wednesday, we'll pick up with processing. And so these get processed. As you can imagine, the split genes get processed differently than the ones that don't have introns. See you on Wednesday.
Having a hard time seeing if you can change my prescription. And the last
Or sit
exam
closer to the front like I do.
I should, but sometimes it's hard. I'm going to. Can I take a picture, please?
Sure.
Yeah, well, I'm going to take a picture as well.
Picture for
I
posterity.
ran out of contacts in my glasses and I pulled prescription, so I'm like, I can't see.
Yeah, I got an exam, and this time I need to go back. It's not good. I can tell. Really?
Taking pictures now? We were talking
about a cancer where it comes from using like percentages? Would those be only percentages that work? Or
Yes, and because everything else is spontaneous.
okay, the rest is just like
Yes, exactly. Those are the three numbers.
those are the three.
Thank you.
Yeah, so most cancers have no known origin, just spontaneous, random.
And then even.